Let's talk about brains. You've worked your brain pretty hard already today, and it's time to look inside that gray box. And let's start with some big questions. What makes you think and love and talk and play chess? What makes you tick? And what makes each of us so very different? We've seen tremendous diversity in the preceding speakers today. And what makes us unique as a species and as individuals? Well, it's all in the connections, the wiring of your brains. And by that, I mean the physical components, the neurons, axons, dendrites, and synapses that we'll learn more about. And I also mean the strength of connections, what we call functional connectivity. Let's take a quick look at the parts list inside your skull. I love numbers, so you have a squishy brain that's about 3 pounds in weight, and it contains about 90 billion neurons or nerve cells. The dominant component of our brains and what makes us unique is the cerebral cortex. It contains only a minority of the total number of cells, but that's because it's chock-a-block full of the synapses that do the information processing and computation. There's also about 100,000 miles of wiring, the cabling in the underlying white matter. Underneath the cerebral cortex, underneath the cerebral cortex is the pint-sized cerebellum that contains the great majority of your neuronal count, and it's responsible for coordinating your movements and your thought processes. Then there's a third collection of subcortical nuclei, bundles of neurons in the central core of the brain that are small in total number, but are incredibly important for keeping you alive and well. The brain is often compared to a computer, and the comparison, the analogy is apt in many respects, but there are profound differences, starting with what neurons look like. Each nerve cell has an antenna-like set of processes or appendages we call dendrites. It has a single process that we call an axon that goes some distance, then branches profusely and contacts many target neurons through synapses made on their dendrites. Each neuron has about 10,000 synaptic connections, and it conveys information through electrical signals that we call nerve impulses. And each neuron crackles away at a rate of about 1 to 100 nerve impulses per second. So if we could listen into that, you'd hear the music of a neuron on its own. That's the message working within your brain. This is very different from how computers work. Computers and their electronic components use electrical signals, but they process information at vastly higher rates, are able to carry out literally billions of calculations per second within each little chip. And that kind of processing power is what allowed IBM's Deep Blue computer to beat Garry Kasparov in the World Chess Championship nearly 2 decades ago. Sorry about that, Susan. But we know that computers are getting more and more powerful year by year, and yet we pride ourselves in asserting and believing that our brains, we are smarter in many ways, we're more clever. And now we want to ask why. Well, in some sense, it's because we're wired better, we're better connected, but how can we explain that in more detail? We do not know the answer. I won't pretend to give you full answers, but we'll explore this by looking at connectomes. So what is a connectome? It is a, quote, comprehensive mapping or charting of neuronal connections. But I put it in quotes intentionally because we have to look at and think about connectomes in 2 very different ways. One is the macroconnectome, macroscopic, because we're looking at long-distance connections from one patch of gray matter to another through the underlying white matter, but we can only do that down to the level of the individual neuroimaging volume elements. We call them voxels, and they're about 1/10 of an inch, millimeter or two on a side, and each of those voxels contains hundreds of thousands of neurons. So that's the limits of macroconnectomics. And when we switch over on the right to the world of the microconnectome, the domain of using electron microscopic images to reconstruct the location and identity of every single neuron, synapse, axon, and dendrite, but only over a limited volume that is still smaller than one of those neuroimaging brain voxels. So for the rest of this talk, I'm going to focus on the human macroconnectome, and to chart it, we need good maps. And that brings me to another fresh analogy, that of cartography. I grew up in a world where I learned about the Earth and what's on its surface through book atlases and looking at page after page of what was interesting portrayed on maps of the Earth's surface. Little did I realize that I would spend most of my professional career making maps of the cerebral cortex in various species. First, monkeys. You see here a vintage 2-decade-old map of the monkey's cerebral cortex that kind of lays the landscape. It shows in different colors little patches that represent cortical areas in the monkey, and they're analogous to the political subdivisions that fill the landscape of the Earth's surface. We also made maps and charts of brain connections, and you see on the right a layout, a subway chart kind of description of hundreds of pathways interlinking dozens of cortical areas associated just with the sense of vision in the monkey. Now that was progress, but it was frustrating at the time in 1991 that we knew virtually nothing comparable about the human brain. Fortunately, and I'm here today because in the ensuing decades there's been just tremendous progress in developing new methods and generating torrents of data that inform us about human brain circuits. So I'm going to talk about the Human Connectome project, give you a quick overview. We're going to look a little more closely at the inner workings of brain structure. I'll show you how we can map brain activity and brain circuits using what we call functional MRI, and then I'll give you a quick glimpse about individual variability and the heritability of brain circuits. So the Human Connectome project aspires, and we're about halfway through, studying brain circuits and its relation to behavior in a population of 1,200 healthy adults, all born in the state of Missouri, and all parts of families that have twins and their non-twin brothers and sisters. We're using cutting-edge methods of gathering and analyzing neuroimaging data, and we're already sharing terabytes worth of data, that's a lot, with the scientific community and trying to accelerate discovery science. We also anticipate that this information will be valuable as a baseline, and the methods will help accelerate studies of brain disorders that afflict humanity all too seriously. When I say we, I mean a really fabulous team of greater than 100 investigators and staff members concentrated at Washington University, but including wonderful colleagues at 9 other institutions, including our partner institution, St. Louis University. So what are we learning and how do we do it? Let's start with a glimpse at a slice through the human brain, and you can see the outer rim of the ribbon of cortical gray matter, and we want to capture the shape of that gray matter sheet, and we'll take a scroll through part of the brain as we slice through, and you can see the blue and green contours are capturing the shape of these very complex cortical folds. And we can turn this slice-by-slice movie into a computerized reconstruction, a brain model that will take a spin around, and you can see these shallow and deep folds, the convolutions peppering the cortical surface. But we can now smooth, in the computer, we can smooth out and inflate the brain like a balloon, and we can preserve the underlying shape. The darker it is on this inflated surface, the deeper it was in the original 3D model. We can do this for the left and right hemispheres combined, put them side by side, and use this as an anatomical substrate for visualizing brain activity. So on the right is the pattern of brain activity revealed by functional MRI in one moment in one of our participants' lives while she was lying in the scanner at Washington University. This fMRI signal reflects ongoing brain activity. The yellow and red regions, patches, are where neural activity is high. The green and blue patches, neural activity is low, it's cooler for the moment. But your brain and her brain are active all the time, literally 24/7. It's this pattern of activity that drives our thoughts, our dreams, what we do. And we can now look at a movie of this participant's life in the 40 seconds in the scanner, and you can see that there's a complex symphony of brain activity, changes in space and time. And we can zoom or look closely at a couple of patches. Locations 1 and 2 tend to go up and down in activity together. They are correlated, whereas location 3 is not correlated, is even anti-correlated with the others. This pattern of correlations we use to estimate the strength of connections, what we call functional connectivity. And I'll show you more of that, but first I'm going to illustrate another type of information we can gather. The same participants, but in a different scan session, we ask them to do a set of 7 different tasks in the scanner. One of those tasks is really simple. Move your hand back and forth. And in this participant, a region in the left cerebral cortex was controlling the movements of the right hand, and that's the patch shown in red and yellow. And when we do this in 200 subjects, we see a consistent pattern, so we get a smoother, cleaner signal. That's a really simple task. Now let's look at something a little more complicated. I'll show you 5 pictures in succession. And the first one is, they're all tools. The first tool, take a look at it. The second tool is different. The third tool is different. And now I'm challenging you, does the picture number 3 look identical to the 2 back, picture number 1? The answer is no. What about number 4? It doesn't look like number 2. It was actually shown as number 1. Now number 5, you should remember that it looks just like number 3. Whether or not you got the answers right, we're not going to take a full exam on this, but this challenged the portions of your brain involved in what we call working memory. Regions in the front and the back of the brain, shown in yellow and red, are part of that working memory network. That's one kind of thing we're learning. We want to probe this working memory network more closely by asking how are these regions connected to one another and to other portions of the brain. So we use this method of resting state functional connectivity that I illustrated a few slides back, and we interrogate location A up here and ask who's fluctuating in resting state neural signals, and the answer is the strongest correlations are with other portions of the working memory network. These domains are working in concert in a coordinated way. This is not a fluke. We can take another task, one of lying in the scanner and listening to stories and answering questions about those stories, and that activates a system we call the language network that's in a different region of the cortex, particularly on the left hemisphere. And if we interrogate the language network and ask where is location B communicating most actively, and the answer is a completely different from the working memory network. It's communicating with other parts of the language network. So this gives you a hint, a glimpse, a tip of the iceberg of some of the big questions we in the Connectome project and others in the community who are taking advantage of this resource are probing. We want to understand how many different brain networks and subnetworks can we identify, what does each network contribute to behavior, and how does behavior and brain circuitry co-vary? Are people with better working memory, do they have different networks than those with worse working memory? What about the differences in brain circuits for high versus low IQ? And what about the plague of brain disorders that afflict humankind? We know, for example, that people with schizophrenia have abnormal they suffer from abnormal, pathologically bizarre memories? Can we identify the abnormalities in the working memory circuits in future studies that build on this information and methodology we've developed? And in the last few minutes, I want to touch upon one more issue, what we call nature versus nurture. What aspects of your brain circuits are inherited, given to you by your genes and the instructions that follow, and what aspects are shaped or modeled by your experiences in life. I'll give you just one example that concentrates on the cortical folding patterns that you've already seen illustrated. So in the slide that's visible now, you see 4 different cerebral hemispheres, the right hemisphere, from 4 people. They're 2 pairs of identical twins. Well, if they have the same genetic instructions, you might expect their cortical folding patterns to be identical. They're not, and in fact, if you look more closely at the maps of cortical folding seen on the right side, you're hard-pressed to tell which pair would be the sets of identical twins. I'll just tell you the answer, and in the top you see that people A and B are twins. They have differences aplenty, such as those shown by the red arrows, but there are, in fact, more similarities if you look closely and carefully analyze it. Similarly, people C and D have prominent differences in some regions, but they are more similar than people taken at random. So what do we learn from this? First of all, cortical folding patterns, the feature that gets a lot of cortical turf inside your skull, it's highly variable across all individuals. Identical twins have folding patterns that reveal some heritability, but there's surprisingly high amounts of divergence across identical twins. And we're just getting going in analyzing brain circuits, and I won't illustrate, I'll just assert that brain connections are highly heritable, but they are also strongly influenced by our environment, as you would expect. So what is it that we're learning, to cut to the chase? We're learning a huge amount about brain connectivity and its variability. We're getting insights into what makes us unique as species and as humans and individuals. We're learning about the contributions of genes and their environment, and we're developing and making freely available to the community methods and a baseline resource of information that will inform our abilities to diagnose and better treat diseases and disorders in future studies. And that's hugely important because everyone in this room knows one, probably many people, your friends, your family, who are suffering from schizophrenia, Alzheimer's, ADHD, and autism, and it goes down the line. So I can only spend an extra moment to thank a wonderful team of colleagues at these 10 different institutions who've worked intensely over the past 3.5 years to bring us to this current state of knowledge. And I want to close with one final analogy. There have been revolutions in cartography of both the Earth's surface and mapping of the brain. It all started with individual maps, quaint maps of the Earth's surface, historically charming but inaccurate maps of individual brains. It moved, it graduated to book atlases that had more information and a lot more accurate for both the Earth and the brain. Then there was the explosion driven by computerized imaging technologies that gave us satellite imaging for the Earth and neuroimaging for the brain. Then there was another revolutionary transformation. Google Earth and Google Maps help us navigate the world in ways we couldn't dream of not so long ago. And our hope is that revolutions in connectomics will inform us deeply about the nature and interpretation of human brain connections in health and disease. So thank you for your attention.